Each of our cells contains the instruction book for our whole body, packaged up as DNA. And for the past 3 decades or so, scientists have figured out how to edit that code. They can dig into the actual double helix to fix a genetic mutation and cure illnesses like the immune condition that used to be called bubble boy disease, or reprogram a cell to turn it into, say, a cancer killer. That makes gene editing a profoundly powerful technology. But it's not reaching anywhere near the number of patients who could benefit from it. Doctors have already used gene editing to cure more than 100 people with sickle cell anemia, for example. But almost 8 million people have that disease, and gene editing is way out of reach for the vast majority. That's because it requires much more time, money, and grueling preparation than most people can afford, especially if they live in a developing country. But in Seattle, one lab is chasing a breakthrough that could transform gene editing from a pricey boutique procedure into a widely accessible treatment. I'm Gabriel Spitzer, and this is Pocket Science from KUOW. In each episode, we comb the Pacific Northwest to uncover the fascinating, weird, groundbreaking science happening in our backyard and hear from the scientists figuring it out. Today's episode, innovations in gene editing that could transform how we treat diseases. It starts in a lab at the Fred Hutch Cancer Center with a guy named Hans-Peter Kiem.
I work on therapies for oncology and for genetic diseases.
Dr. Kiem is tall and lanky with bright eyes. He's been at this work since earning his MD and PhD in Germany in the 1980s. The idea of rewriting a person's DNA to cure disease used to be almost unthinkable. Imagine designing a microscopic contraption that could track down the exact right cell that's causing trouble, carefully slip inside without hurting it, find the cell's DNA and scan the 3 billion or so units of code it contains, and then spot a typo and fix it? It's a pretty daunting engineering problem. But that dream led to the first big breakthrough in making gene editing a reality. Starting in the 1970s, researchers figured out that nature had already done a lot of the work for them by creating natural experts at manipulating genes. We're talking about viruses.
It really starts with leukemia viruses that were discovered, many, many years ago now. And we've learned that these viruses can get into the cell and then easily integrate into the cell.
Viruses have evolved a whole bag of tricks for reprogramming our cells to turn them into virus factories.
So they've really learned how to get into the nucleus of the cell and integrate into the genome, which is the critical part. Viruses have done this for millions of years.
Viruses are master infiltrators. But what if instead of delivering a poison pill that makes you sick, they could deliver something helpful? So scientists learned to modify viruses by stripping out everything that causes disease, but still leaving their ability to sneak into cells and find DNA. They essentially became delivery devices for the gene therapy. The scientists call them viral vectors.
The difference between a virus and vector is a vector can only infect once, and that's it.
The vector can carry with it a tiny engineered molecular machine, like you might have heard of CRISPR-Cas9, which then does the cutting and pasting work in our DNA. And for diseases caused by a simple gene mutation, this tech is already a game changer.
The main interest has been currently on sickle cell disease and thalassemia because there's the most patients that would benefit from that.
Maybe we could spell out the sickle cell process a little bit. Can you sort of remind us what goes wrong in that disease and how this process would go about correcting it?
Those cells, unfortunately, they clump in the bloodstream, and that causes a lot of pain for patients. It can cause bone pain, it can cause the spleen, also liver and lung, so it can cause a number of problems. But I would say mainly a lot of pain, and that pain happens over and over.
That cell defect is caused by?
Just a mutation, one single mutation.
One little mutation in the billions. Mm-hmm. Scientists have proven that they can fix that mutation with gene editing. There's even an FDA-approved therapy on the market. It works. So why would we need something more?
That's also a very good question. Why do we need something more? So we've learned now we can do it. But it is a very complex process. That means a patient comes in, then we have to collect the cells, bring them to a facility where we can then modify these cells, introduce the correct gene, then these cells will be collected again, shipped back to the hospital where the patient is being treated. And if we have a sufficient number of cells, these cells can then be given back to the patient, but that requires the patient to be in the hospital. So the entire process is usually several months.
That involves a lot of money, a lot of time, very sophisticated institutions, right? It's not going to be available in a place like sub-Saharan Africa.
That's exactly right. It's even hard to deliver it in this country because of the infrastructure required to deliver this treatment.
It's also hard on the patient. Before you can reinfuse a person's modified cells back into their body, they get a brutal dose of chemotherapy. But what if you could skip that brutal part and still get the benefits?
So we've been thinking about this for quite some time. I've been aware of the toxicities, the side effects from the chemotherapy that is required for bone marrow transplantation, and then also now for this kind of gene therapy.
And as anybody who's ever undergone chemo or knows someone who has, it's obvious why you would want to avoid that from a person's point of view, right?
It is grueling, the chemotherapy, and so we've thought a lot about how could we deliver this in a more reasonable and less toxic fashion. And that's where we've started about 15 years ago now, thinking about what if we could modify particles and simply inject them directly into the bloodstream. If we had this sort of simply gene therapy in a syringe, we could simply inject our particles and they would find then the defective cells directly in the body and correct them.
By Kiem's thinking, this could be the breakthrough that puts gene editing within reach of the masses. By doing the whole process inside a person's body, in vivo as they call it, all that work and technology that now lives in big expensive labs would be squished down to fit into a syringe that you could just keep in the freezer. Suddenly, a multi-month stay in the hospital becomes an outpatient procedure. Instead of rounds of chemotherapy, you get a shot, a Band-Aid, and a lollipop.
That's where we make the particles.
In a sterile room packed with biosafety cabinets and freezers, cell cultures sit in incubators. They're injected with specially designed rings of DNA, which cause them to crank out little gene editing machines.
We take the cells out and then pipette in those plasmids to modify the cells, and then those cells will produce these magical particles.
You're giving the cells instructions and using the cells as little factories?
Those are the factories, exactly.
The gene editing package is housed in a hollowed out virus to be deployed inside someone's body for a fully in vivo procedure. I wonder, I'm going to ask you to sort of do something a little weird. If we could imagine for a second that we were to shrink ourselves down to a tiny, tiny size and climb inside that one of those viral particles that you have made into a vehicle for transporting the therapeutic payload or whatever. Can you kind of take us for a ride, like narrate what that would look like if it were a little tiny movie from the point of view of somebody riding inside that virus?
Well, I'd say, this sounds like the magic school bus. I'd say to everybody, get on the bus.
Seatbelts, everyone!
Where are we going, Ms. Frizzle? Now we're heading for the bloodstream.
First step would be, we need to make sure this bus has the right handles or keys on the outside, because that's critical. When that bus gets injected into the bloodstream and floats through the bloodstream, it needs to find the right cells and the right locks on the cells. They're also called receptors.
And let's take a second to talk about that, because your viral particle doesn't have a brain. It doesn't have free will. It's not making decisions about which cells to choose. So how is it making its way to the right cell and then engaging with it?
So we can now, and this has been years of development, we know we can use fragments of antibodies that very specifically recognize receptors on cells. And this is then like the handle or the key and the lock.
That particle is kind of bouncing off of lots of different cells, but when it runs into the right one and the key and the lock happen to knock into each other, they will bind.
They bind. So it's not difficult for them to find the right cells, to be honest, the school bus. It's more important that they avoid the wrong cells. We don't want to have this off target.
Once we bind to the right cell, the ancient viral programming takes over, and the bus slips inside.
The virus has developed the ability to get into the cell. We still use that function and that property from the virus.
Our school bus has made its way through the envelope of the cell. Now we're inside and you can see all that cytoplasm and all that stuff people remember from their biology classes. Then what happens next?
So the next big step, of course, is we need to get into the nucleus. That's the next part. And then, of course, integrate our information into that big instruction book.
All right, so once our school bus has opened up its little box of tricks and it has unzipped the double helix and it has inserted its page and then I assume kind of put everything back together?
Then the cell has new instructions how to make that particular protein to fix sickle cell disease or other genetic diseases.
How close are we to this being generally available, and what stands between us and that?
Well, we have to do now a lot of studies in patients.
Those studies are needed to dial in the right dosage, to ensure that the new cells don't get swamped by the defective ones, to make sure that the benefits last. There's still a long to-do list.
We know now it's safe, and it's been done a few times, but it will take several more years to really know how do we need to tweak this now to make it as good as the ex vivo.
Scientists hope that one day this technology could basically wipe out sickle cell anemia and other more rare diseases, as well as fighting cancer and eventually even curing HIV. Dr. Kiem, how long have you been at this?
Well, I've been doing gene therapy for more than 25 years when it started out very early.
I mean, it must be a very different world now than it was when you started.
Oh, it's been fascinating to see the progress. When we see in the lab that cells, we can correct genetic defects, we can fix diseased cells, it's just an amazing feeling, and to be able to almost magically fix that disease.
It must be so interesting to be able to kind of make predictions back in the 90s, say, and then see it through to actual, working therapies and lives saved.
It's been an amazing journey, absolutely, and very fascinating. But it is a journey.
Do you think of this as being a little bit of a holy grail for your own career?
Absolutely. That's exactly. When I get up, this is what I think. I do think this is what I want to make sure we can accomplish. This is what I would like to do with the remaining time I have in the lab, and to really develop in vivo-based therapies for cancer and for genetic diseases. Well,
thank you.
Of course, thank you very much for coming by.
Thanks for being our bus driver.
Yeah.
Hans-Peter Kiem is a professor at the Fred Hutch Cancer Center and Stephanus Family Endowed Chair for Cell and Gene Therapy. You've been listening to Pocket Science from KUOW. I'm Gabriel Spitzer. This podcast was edited by Jim Gates and produced by Shane Mailing, music by Artlist and Blue Dot Sessions. And if you've enjoyed Pocket Science, tell a friend or write us a review. It really helps a lot. And you can support our podcast by visiting kuow.org/pocket. We'll see you in 2 weeks for another episode of Pocket Science.